Welcome to my channel and Pure Biology. And today's topic is the structure of tRNA. And before going to my topic, I want to say thanks to all of you who have already subscribed my channel and support me for the growth of my channel. And nobody till now subscribed my channel. Please do subscribe because your support is very important for the growth of my channel. Okay, so let's start. This is the structure of tRNA. First of all, why the tRNA is important? You all know in the DNA there are three processes. One is the replication, another is the transcription, and the last one is the translation. Okay, so replication means the formation of the DNA from another DNA, parent DNA obviously. Transcription means formation of the mRNA from DNA, and the translation means the protein synthesis. Three are very important because without this, the cell cycle that is very important for the cell division. Okay, so these are not completed. So these are very important topic in obviously in our life, you can say, or in the subjects. So today's topic is the tRNA structure. I just want to say briefly what kind of structure of tRNA you can see, because tRNA is very important for the translation process, and I obviously described in my channel explain that the transcription process and the translation process, but slowly. Okay, because these are very vast syllabus. Okay, now you can see this is the rough structure of the tRNA. Okay, so this tRNA actually that is very important for the translation. Each contains stem and loops. These are all the loops. You can see these are all the loops, and these are the stems. Okay, stems and the loops. So generally tRNA, this is 75 to 90 nucleotide long, and each type having the different sequence by which one tRNA obviously separated from another tRNA due to the different sequence, because one tRNA has a sequence is not similar to the other tRNA. And another way I can say you that the differences in the nucleotide sequences in actually the ability, it is the ability of the particular tRNA molecule to bind a specific amino acid. That means its process or binding process with the amino acid depend generally this nucleotide sequence. This nucleotide you can see here. These nucleotide sequences are very important for each and every tRNA, because depending on this, which amino acid bind to what tRNA, that depends. Clear? So, and you can see this is the structure just like clover leaf. It is a structure just like clover leaf. Now, which stem or which loop is known what? Okay, sorry for the disturbance. So, you can see this one is the acceptor arm. This is the acceptor arm. Sorry. This is the acceptor arm. This is the acceptor arm, and this is the site for amino acid attachment. This is the site for amino acid attachment. Here the amino acid generally bind. This is the site for amino acid attachment, and this is the acceptor arm. This one. Second, this is the D loop. This one is the D loop, and this is the D stem. And this loop is called the anticodon loop. Okay, anticodon loop, and three are there, so form the triplet. Three nucleotide sequence. You can see base here or bases. You can see that three nucleotides that form the triplet. Okay, and this is the anticodon loop. This is called the variable arm. Okay, this is called the variable arm, and this one is the T loop. Okay, so what is saying here, the acceptor arm has the 7 base pair. You can see this is the 7 base pair. 1, 2, 3, 4, 5, 6, 7. The 7 base pairs are there. including the conserved CCA sequence, and you can see here from here to this portion is the CCA sequence. Here this portion is the CC. This is the ACCAC conserved sequence is present. Next one, the D stem and the D loop that are variable length, and the anticodon stem has the 5 base pairs, but loop has 7 nucleotides. Okay. So anticodon, which one is the anticodon? This is changes. What is saying here? The loop are variable length. Loop and stem. Obviously, the stem and the loop are the variable length. But anticodon stem has 5 base pairs. 1, 2, 3, 4, 5. 5 base pairs, and loop has 7 nucleotides. 1, 2, 3, 4, 5, 6, 7. Okay, 5, 6, 7. Or 7 here, 1 thing, sorry. Okay, so this is 7 base pair. 1, 2, 3, 4, 5, 6, 7. 7. This one is the 7. So this is changing, or the length of D loop and the D stem that changes, or they are generally in the variable length, but the anticodon, loop and stem, they have generally, this one contains the 5 base pairs, and this contains, the loop contains the 7 nucleotides. Okay. The variable arm has 4 to 5 nucleotides but can contain 24. This is called the variable arm. This one is called the variable arm, and it contains generally 4 to 5 nucleotides. 4 to 5. 1, 2, 3, 4, 5. 4 to 5 nucleotides are there, but it has the capacity to contain 24 nucleotides. Clear? The T stem has 5 base pair. This is the T stem and this is the T loop. So T stem that has 5 base pairs, but loop has 7 nucleotides. You can count there. 7 nucleotides are there. 1, 2, 3, 4, 5, 6, 7. 7 nucleotides. And these are thought to fold L-shaped or 3D structure, 3-dimensional structure. So this is if you it is seen in the 3-dimensionally, it looks like the L-shaped structure. The tRNA looks like the L-shaped structure. Now we can briefly know how the tRNAs are very important for binding of the amino acid. And here the important role is seen in ATP. Okay. It is very important role. First, the amino acid and ATP bind to the specific amino acyl tRNA synthetase. Okay, this is A for amino acyl tRNA synthetase. Okay, so first what happened? The amino acid and ATP both bind to the amino acyl tRNA synthetase. What happened after that? This ATP is hydrolyzed to form AMP. ATP hydrolyzed to form AMP. That means and here in that situation, the AMP joined to the amino acid as AMP and to form amino acyl AMP to the tRNA. Sorry. So what happened, you can see here, the amino acid and tRNA bind to the amino acyl tRNA synthetase. And what happened in that situation? The ATP hydrolyzed to form AMP. And here what product is formed? Amino acyl AMP tRNA. And after that AMP displaces from this compound and amino acyl tRNA that released from this compound. This one released amino acyl tRNA and AMP also released or displaces. And in that situation, amino acid bind to the, actually so there are 3 steps you can see. What are the 3 steps? First, amino acid and ATP both bind to amino acyl tRNA synthetase. This is enzyme. And after that, the ATP hydrolyzed to form the AMP. And here what happened? This AMP bind to the amino acyl tRNA. And the structure, the formation or the product, you can see the amino acyl AMP tRNA. Okay, amino acyl AMP tRNA, and after that this amino acid transferase amino acid tRNA that release from AMP. Okay, and AMP also displaces from this product. In this way, the amino acid bind to the tRNA. Okay, so here actually amino acid bind where? What is the location? The OH of the tRNA 3' end. Actually, it binds generally the covalent with the bonding form, that is the covalent linkage, and it is seen between the carboxyl group of the amino acid and the 3' hydroxyl or 2' hydroxyl group of the ribose of the adenine nucleotide. Okay, so what the covalent bond is formed? The covalent linkage is formed generally the hydroxyl group, 3' hydroxyl group or the 2' hydroxyl group of the ribose of adenine. Okay, ribose of adenine nucleotide, and this is found actually here. The carboxyl group is also important. Okay, the carboxyl group is amino acid. The amino acids carboxyl group COOH. It is very important for the binding. Okay, COOH group. And the hydroxyl group of the ribose of the adenine. So, amino acid COOH group and the adenines OH group. Clear? Adenines OH group and amino acid COOH group is very important because there the covalent bond is formed. Generally, you just learn that amino acid bind to the OH group, bond or OH group obviously of the tRNA at 3' end. So this is the structure of the tRNA. Today I explain you, and I think you can understand it properly. And in the next video, I will explain you the transcription process and transcription enzymes obviously, and slowly I will explain you the translation. Okay, so today's topic is now completed here, and thank you to all of you.